Then, hello everyone. So welcome to this ISV Next Gen webinar for the month of April, a bit later than what we usually have, but with two exciting speakers. So Associate Professor Dr. Kanitha Patarakul from Chulalongkorn University in Thailand and Alcidia Ramos Barros from the University of Geneva. But first, as usual, let me present to you a little bit who we are and what we do. So first of all, I would like to introduce you or push you to subscribe to our annual congress that will take place in Antwerp. The 12th and 14th of October 2026 will be very exciting. You will have a lot of exciting speakers. Global Expert will be a great occasion to network and to meet global experts. So here you have some information on the abstract. Of course, we would like you to submit one. The deadline is the 12th of June. You have a whole variety of different topics on vaccinology, so please submit it. And you will be also in the competition for the Bright Spark. So there are a lot of nice prizes for best poster and oral presentation. So don't hesitate to participate. Just a small reminder that the early bird registration deadline is for the 31st of July. So please go ahead. For our Mother Society, the next webinar will be the 13th of May 2026, of course, at 14 Central European time. We will have Nathalie Garçon. So please, it's going to be a very interesting webinar. So please don't hesitate to participate. This is just a review of our annual supporters. If you want to join us or follow us, please don't hesitate to follow us on social media. Here are some QR codes to facilitate your entry. So don't forget that it's very easy to join us and to become part of our group, especially we are next gen, so especially for us, it is very cheap. As a postdoc or PhD students, the fee is $35 per year. So don't hesitate to join us. Our next webinar will actually be very soon, the 7th of May. We will have also two exciting speakers, so Dr. Rory De Vries from the Erasmus University and Arturo Liñán Torres from Universidad Nacional Autónoma de Mexico. So let's go back to us and to today's webinar. So I would like to introduce you Dr. Kanitha Patarakul. Hello, Kanitha. Thank you very much for being here. So I will just give a very brief background of Kanitha because she will present us also her background. So she's currently the chair of the Department of Microbiology at the Faculty of Medicine at Chulalongkorn University and head of the Center of Excellence in Vaccine Research and Development at Chula VRC. She's also chair of Institutional Biosafety Committee of the Faculty of Medicine at Chulalongkorn University. So for the rest, I let Kanitha directly present herself. As a small reminder, if you want, we will have Kanitha presenting, then we will have a couple of burning questions if times allow, and at the end we will have a Q&A session. So in case you have any question, don't hesitate to write them in the Q&A below. Kanitha, the floor is yours.
Thank you so much for your introduction, and thanks again for inviting me to join this webinar series. Good morning or afternoon, everyone. I would like to share my research work on leptospirosis vaccine developments. Lesson learned from protein subunit vaccine to mRNA vaccine. Just a brief background of myself. After I trained as a medical doctor at Chulalongkorn University and then pursue my PhD in microbiology at Georgetown University and work on Borrelia burgdorferi, which is another spirochete. And when I graduate and become a faculty member at Faculty of Medicine, Chulalongkorn University, at that time there's an outbreak of leptospirosis, and there's no Lyme disease in Thailand. So that's why I shift my work on leptospirosis instead, and then joined Jula Vaccine Research Center for vaccine development. Today, I will focus my talk on the rationale and evolution of leptospirosis vaccine development, identify the key antigens and vaccine candidates, and some of the results of recombinant protein subunit vaccine and mRNA vaccine platforms that I use. And I test it in animals models in mice and hamsters. Leptospirosis is one of the most widespread zoonotic disease worldwide. It's caused at least 1 million cases and approximately 60,000 deaths each year. The burden is especially high in tropical and subtropical regions. For vaccine development, this is important because leptospirosis is not only a human health problem, but also a One Health problem involving animals, environment, and occupational exposure. The causative agent of leptospirosis is bacteria, is a leptospira, is a thin motile helical spirochete that has the double membrane similar to gram-negative bacteria and has periplasmic flagella. There are more than 300 serovars and more than 20 genome species, and the dominant serovars vary by geographic area. This diversity makes vaccine design challenging because protection against one serovar may not provide broad protection against others. Leptospirosis is transmitted through skin abrasions or mucosal surface after exposure to contaminated water or soils or urine of infected animals. Humans become accidental hosts, and after bacteria entry into the body is disseminated through the bloodstream and cause acute phase infection. Patients have fever. And then leptospira reach target organs, multiple organs, including kidney, liver, lung, and can cause multi-organ dysfunction that can be fatal. The prevention of transmission by hygienic control might not be sufficient. So leptospirosis vaccine is required, especially for high-risk group. Currently available leptospirosis vaccine are inactivated whole cell vaccines or Bactrians. And there's a limitation because it can confer short-term immunity, and it can protect against only limited heterologous serovars. Usually, it's only the serovar that are component of the vaccine and can cause potential side effect. So it has been used mainly in animals. So that leads to development of recombinant protein subunit vaccine to overcome the limitation of bacterians. So usually this vaccine contain conserved outer membrane protein for cross protection and hopefully have fewer side effects. The subunit vaccines can simulate both humoral and cell-mediated immunity and induce memory cells, so it can confer longer immunity than the bacterians. There are several vaccine candidates has been tested. The promising one is the surface exposed. There are surface exposed protein called leptospiral immunoglobulin-like protein, LIG-A and LIG-B. And this protein has been used in different studies. For example, the vaccine has been used either C-terminal or N-terminal part of the vaccine, and the results can vary. This is the survival efficacy of protection. The difference may be because of different fragments that have been used, the adjuvant, the routes, and the dose for challenge, and also the models that use. So there will be still a room of improvement. In my work, my team that we study using that with the factor H binding protein, because leptospira are resistant, pathogenic leptospira resist complement killing in the blood. So because it, for example, it can bind to complement regulator such as factor H. So when the components of alternative pathway try to deposit the native pathogenic leptospira, it can be inactivated and make the bacteria resist complement killing. And LIG-A is one of the factor H binding protein, but there's still several factor H binding protein on the membrane of leptospira. So one monovalent recombinant subunit might not be enough. So we combine measure for outer membrane protein that are factor H binding proteins, and adjuvant with the liposome-based adjuvant that we collaborate with the VFL Switzerland, and then immunize this vaccine formulation 3 times in hamster 2 weeks apart, and then challenge the hamster with violent leptospira. And unfortunately, the combination of 4 antigen still get the survival similar, 60% survival similar to the monovalent LIG-A C alone. However, you can see that when we look at in the kidney tissue, we found that combination of the 4 antigen can reduce leptospiral burden in the kidney. So this is the important, because not the binding of the bacterial surface is important, but interfering with the immune evasion mechanism might help to improve the vaccine efficacy. The recombinant protein have its own limitation. As I told you, that they require adjuvants. And if we need multiple antigens to induce complete protection, to produce the protein at large scale is quite difficult to get a high yield, and have the problem with solubility and the quality of the recombinant protein. So that's why we ask if whether the mRNA vaccine platform could improve leptospirosis vaccine development, because it can induce high immunogenicity and can be stable with the lipid nanoparticle and easy for the large-scale production, especially when we want to combine multiple mRNA constructs. So we collaborate with Professor Drew Weissman at University of Pennsylvania to use the nucleoside modified mRNA that can induce both humoral and cellular immunity. The most important protective immunity for leptospirosis is the humoral immunity, which is antibody. But T cell is still important to help that influence the magnitude and quality of antibody response. This is our work that just recently published, and this is the first mRNA vaccine for leptospirosis. And we use leak protein as the model to show the efficacy of the mRNA vaccine. This is the overall concept of the study. After we make the mRNA vaccine construct and encapsulate with the lipid nanoparticle, we'll check the expression and secretion in vivo in the cell lines and check immunogenicity and protective efficacy in animals. This is the design of the mRNA construct. We use C-terminus of lead A and N-terminus of lead B. And this one, when we design, it has the either native sequence, the same as in Leptospira, and also optimize the codon usage so it will be different in nucleotide level, but has the same amino acid sequence of the protein. And we use these two versions and transform into the plasmid, put into the plasmid, and then do the IVT and make the mRNA construct. The mRNA construct also contains signal peptide, which is the human synthetic signal peptide, so it can be expressed and secreted in mammalian cells. And this mRNA construct are encapsulated with lipid nanoparticle that's a pattern of Geneva. The mRNA-LNP were transfected into HEC293 cells, and then we check the protein expression and secretion by SDH-PET and Western blot. You can see here that in the culture supernatant of both lead A, C, and lead B, N, we show that after 48 hours expression, the protein are secreted into the culture supernatant, and the optimized codon construct get higher expression than the native ones, as expected. However, the protein size is bigger than what we expected. This is to compare with the recombinant protein that's made in E. coli. So we check with the glycosylation. So after the glycosylation treatment, you can see that the protein shift to lower molecular weight. So this protein, they are bacterial proteins and expressed in mammalian cells. So it's underwent post-translational glycosylation. Glycosylation may help in secretion or stability, but it might affect the cons that it can change antigen conformation or mask protective epitopes. But still, we don't know if there's a pros and cons in this case. So we continue to test immunogenicity of these constructs in mice. We use either native or codon optimized construct and immunize mice. This is the JCL-ICR outbreak mice twice at 3 weeks apart, and then check antibody titer by ELISA. And you can see here that during the first immunization, the deep blue one, this is the optimized codon, gets significantly higher than the native codon of the mRNA construct. And these two are better than the recombinant protein. When we immunize in the second time, The second immunization can enhance antibody titer for all vaccine formulation, but still the optimized codon now they have a similar antibody titer to the native codon, but still a little bit better than the recombinant construct. And these antibody titers, they are only have the binding activity, but we want to show if they have functional activity as well. So we use immune sera of vaccinated mice and use it to check serum bactericidal assay to show that, as you can see here, when we incubate the serum with complement serum and with the pathogenic leptospira, and we check the survival of the leptospira over time, and we can see that it can enhance complement mediated killing of pathogenic leptospira compared to the pre-immune or mRNA-LNP group. So we want to check the protective efficacy. So we need to check in hamster model because mice are resistant to leptospiral infection. We use female golden siren hamster, which is highly susceptible to acute leptospirosis. In this case, since codon optimized construct are better than native ones, so we only use codon optimized LIG-AC or LIG-BN mRNA construct. And we use 5 microgram and inject intradermal, or use 1 microgram to inject 5 microgram for intramuscular and 1 microgram for intradermal. So they are using either LIG-AC or LIG-BN in different dose and route compared to mRNA-LNP, which is negative control, and kill vaccine homologous zeroas as a positive control. And we immunize manually the intramuscular or intradermal 2 times, 3 weeks apart, and then challenge with violent leptospira. After challenge, we monitor survival and apply human endpoint criteria. We also collected kidneys for histopathology and qPCR analysis of leptospiral burden. This is the slide to show the antibody levels. And you can see that after the second immunization, we get higher antibody titer for both LIG-AC and LIG-BN. But in our tested condition, intramuscular is better than intradermal. You see here. This may be because of the dose, or we haven't used delivery device that haven't been optimized yet. When we look at the IgG subclass, is a mix of IgG1 and IgG2, not IgG3. So this means that the vaccine can induce both T helper 1 and T helper 2. And we check for the protective efficacy in terms of survival. And you can see that only killed kill vaccine, which is homologous zeroas, can confer 100% protection. But the mRNA, either LIG-A and LIG-B, confer partial protection, but it's not significant different from the mRNA-LNP. And so also look at the protective efficacy in hamster. Look at the kidney pathology and leptospiral burden in kidney tissue. You can see that the kill vaccine can reduce histopathology score and leptospiral burden. But in our group, some group, for example here, LIC-AC mRNA, can reduce some of the histopathology score. However, only survival hamster can show the lower histopathology score and lower leptospiral burden. So in this case, this means the protection is only partial protection and cannot cause a sterile immunity. So to summarize our main finding, that first, leak mRNA-LNP formulation can induce strong antibody response in both mice and hamster. When we use mouse antibody, we can show that it has functional complement-mediated bactericidal activity. However, when we use this construct to determine protective efficacy in hamster, it can confer only partial protection. And the best one that we can get is for LIC-B is 25% survival. So we need to improve mRNA vaccine efficacy. And there's many directions that we can look at. First is the glycosylation site modification that might affect protective epitope. We have to optimize the dose, the route, especially if we want to use intradermal, maybe we need to have effective delivery equipment. And as I talked earlier, that one antigen might not be enough. So we plan to use multiple vaccine candidates to make multivalent formulations. And maybe heterologous prime boost strategy might improve vaccine efficacy as well. So I would like to thank collaborators. Professor Kiet Raklung-Tham, he's the founder of Chula Vaccine Research Center. Professor Drew Weissman and his team at the UPenn, and my team members in the lab, and Chula Vaccine Research team members. Our collaborator for the mouse and hamster study, and LNP, and also granting agency. Thank you very much for your attention. I will be happy to take questions. Thank you.
Thank you so much, Kanitha. It was very interesting. It's beautiful to see how you could proceed, and also this change from proteins to mRNA. This is very, very interesting. I had a question regarding the, maybe we will do just one question and then proceed with Alcidia. Regarding the intradermal, so you said that you're looking for different options of intradermal administration. So were you thinking about microarray patches? What in particular? For the moment, you're just doing an injections, that's right?
That's for intradermal. Actually, the delivery, we have problem with the delivery of the needle-free injection. So that's why we need to use manual injection at that time when we have the experiment. But right now we have a needle-free injection, which is might be get better or enhance immunity better. Yeah, hopefully, yes.
Very much looking forward to see the results from this application. The background, Alcidia is a pharmacist and she's from the University of Geneva, currently doing a PhD in the lab of Professor Gerrit Borchert. So Alcidia, please, we're very interested to hear about your study.
Thank you, Allegra, for the kind introduction, and it's my pleasure to share with you a part of my thesis research, which is ongoing. I hope you can all see my screen. Yes, now, and I will put it in presenter mode. Okay, so I will share with you a part of my thesis research. Guessing the slide is not going. Yes, it's coming. So in this presentation entitled Towards a Tetravalent mRNA-LNP vaccine formulation against dengue fever, I will give you the background and the goals of the project, as well as the methodology used for this specific step and some of the preliminary results obtained, and the perspectives for this ongoing work. As mentioned in the title, we are focusing on developing a vaccine against dengue fever, which is considered by the WHO as the most prevalent and rapidly spreading arthropod-borne viral disease in the world. It is caused by the dengue virus, for which there exists 4 different serotypes, DENV 1 to 4. And the disease is mainly found in subtropical and tropical regions, and nearly half of the world's population is at risk. It is transmitted, so the dengue virus, each serotype, is transmitted to humans upon the bite of Aedes mosquitoes, specifically the Aedes aegypti species. And there has been a steady increase in the number of cases reported to the WHO in the last century, going from 500,000 cases in the year 2000 to over 14 million in 2024, and more than 12,000 dengue-related deaths. And this rapid spreading is one of the reasons why the WHO has considered dengue fever to be one of the top 10 threats to global health in 2019. And as I mentioned, the disease is caused by the dengue virus, for which there exist 4 different serotypes, and each of them is capable of causing the infection and eliciting a specific immune response. As of now, there is no specific antiviral treatment. There have been 2 approved live attenuated vaccines on the market, Dengvaxia from Sanofi Pasteur and Qdenga from Takeda. But they suffer from restrictions of use according to either the age, the serostatus of the patient, and or the category of patients, simply also because there are live attenuated vaccines which cannot be delivered to, for example, pregnant women or immunocompromised patients. In particular, for Dengvaxia, the restrictions of use were due to severe adverse effects observed after commercialization. This is why there is still a need for a vaccine that can be administered to all patients. And our project, the DENVAC project, aims to contribute to ongoing efforts in developing a tetravalent vaccine against dengue fever that can be administered to all. It is an international collaboration between the groups of Professors Ketloy and Prompechara at Chula VRC in Chulalongkorn University in Bangkok, Thailand, and the biopharmacy research group led by my supervisor, Professor Barros, at the Institute of Pharmaceutical Sciences of Western Switzerland in Geneva. So at Chula VRC, our collaborators engineer and manufacture mRNA constructs for each DENV serotype, aimed at inducing potent neutralizing antibody levels. And as formulation scientists here in Geneva, we formulate the mRNA-LNP vaccines and characterize them physicochemically and biologically. The overall aim is to come up with this tetravalent mRNA-LNP vaccine for intramuscular delivery. We would like the vaccine to be highly immunogenic, so capable of eliciting high levels of neutralizing antibodies and specific T-cell responses against all 4 DENV serotypes. A vaccine administrable to all patients, easily manufactured, cost-effective, and possibly stored at fridge temperature. To reach this goal in this step of the project, we have chosen to go with a step-by-step co-encapsulation approach, which was divided into 2 main parts. The first one consisted in a preliminary screening of monovalent and multivalent formulations, And this work was mainly conducted by my former master's student, Tatiana Borges da Costa, where the idea was to encapsulate all 4-denv mRNA, illustrated here, into an LNP formulation, which we called the CL2 formulation. It is a rather classical LNP formulation composed of the 4 types of lipids, which you may know. So a cationic ionizable lipid, a helper lipid, cholesterol, and pegylated lipid at fixed molar ratios and fixed NP ratios. So not only did my former master student formulate this vaccine, but also she physicochemically and biologically characterized all of these formulations and compared between their liquid and freeze-dried forms using a pre-established freeze-drying process that we had in the lab. And in a second step, we refined the tetravalent formulation by adjusting the mRNA dose, upscaling to microfluidics manufacture, and sterilizing the formulations. The CL2 formulation is a formulation that we had previously developed in the lab and used to encapsulate a model luciferase mRNA, as illustrated here. And upon transfecting it to raw cells, we saw high levels of bioluminescence at the 2 used concentrations, so 2 dilutions, here and also here, before freeze drying, so in liquid forms, and also after freeze drying. So you can see that the bioluminescence level are significantly higher than the blank ones here. So these were encouraging results telling us that the CL2 formulation was able to preserve the mRNA integrity and transfection efficacy of the luciferase mRNA after freeze drying. And we were confident to then use it as a delivery platform also for our dengue mRNA. And this is exactly what was done in this preliminary screening step, where monovalent CL2 formulations were manufactured, encapsulating each one of the dengue serotype, as you can see here. So dengue 1, 2, 3, and 4. And we also explored multivalent formulations, so a bivalent one with dengue 1 and 2, trivalent one encapsulating dengue 1, 2, and 3, and a tetravalent one encapsulating all of the 4 dengue serotypes. And of course, we always had the blank formulation as a reference. These formulations were manufactured by ethanol injection method, and size exclusion chromatography was used for ethanol removal and buffer exchange. And as we were freeze drying, a cryoprotectant was added to the formulation before that step. When it came to the physicochemical characterization, we assessed rather classical criteria in the field to ensure that the quality target product profile was met and that the characteristics were compatible with an intramuscular delivery. So we assessed the size in terms of C average, the polydispersity index by DLS, both of them, so dynamic light scattering, and we also checked the zeta potential by electrophoretic light scattering. All 3 criteria were assessed using the same zetasizer. We also checked the morphology of the particles using transmission electron microscopy. We assessed pH and osmolality to make sure that they were compatible with an intramuscular delivery. And we also looked at the concentration of the encapsulated mRNA and the mRNA encapsulation efficiency using a ribogreen assay. When it came to biological characterization, we checked the transfection efficacy using flow cytometry and cytotoxicity using WSC1 and propylium iodide assays. So in the preliminary screening step, we saw for the average and just get my pointer again. Here, we saw that for the average NPDI for all formulations in both liquid and freeze-dried forms, we were within the target values except for a couple of individual replicates. So we had the averages globally below 200 nanometers and PDIs smaller or equal to 0.3. And we saw no real difference between liquid and freeze-dried samples. For some of the formulation, we saw a trend of an increase in the size after freeze-drying, for example, for the blank or the equivalent one, but for the others, it was the contrary. When it came to zeta potential, we had positive values, and this was coherent with the use of positively charged lipids, and we saw pH values that were compatible with an intramuscular delivery. For the osmolality, we saw a very clear trend where liquid formulations had osmolality values around 300 milliosmol per kilo, where freeze-dried ones had values which were the double of that. And this can be easily explained by the use of the cryoprotectant in the freeze-dried formulations. And in either cases, the values were compatible with an intramuscular delivery. When it came to the ribogreen assay for the monovalent formulations, which you can see in the first half, we saw that the CL2 formulation encapsulated each dengue mRNA in a quite similar fashion with encapsulated mRNA concentrations that are alike across the four monovalent formulations. And when it came to the multivalent ones, in terms of the encapsulated amounts of mRNA or concentrations, we saw the linear increase that was expected. Nonetheless, there is a high variability between replicates for the multivalent formulations, which can be attributed to either the poor reproducibility of the ethanol injection method or also potential degradation of the mRNA during the manufacturing process. This being said, by checking the encapsulated mRNA concentrations and comparing it to the initial theoretical one, we knew that for each of the multivalent formulations, we had encapsulated the number of mRNA serotypes that we wanted, meaning the 2 mRNA serotypes for the bivalent, the 3 for the trivalent, and the 4 for the tetravalent here. In terms of the morphology, this was done only on the blank formulation here and the multivalent ones, so the bivalent, trivalent, and tetravalent in liquid forms in the top row and freeze-dried forms in the lower one. We saw the spherical particles that we were aiming to see, and we also saw that the particles diameters were below the 200 nanometer threshold, confirming what we had with DLS, and we saw a bit more aggregation in for the freeze-dried formulations than the liquid ones. For the biological characterization, we checked the transfection efficacy using flow cytometry. So we looked at the mean fluorescence intensity ratio of the formulation over the one of its corresponding blank. And if the ratio was positive, so above 1, we considered the transfection had occurred. Here you can see that it was the case for most of the formulations. We had transfection occurring. For some of them, the liquid forms transfected better than the freeze-dried ones, and for some it was the contrary. Overall, the transfection efficacy is still quite low, and we hypothesize that to be due to the low levels, the low amounts of mRNA that was used in the formulations. So we knew that for the refinement step, we needed to adjust the mRNA dose to reach a higher transfection efficacy. So in this first preliminary preliminary screening round, we managed to formulate the tetravalent Denve-1-4 mRNA-LNP formulation, which, as the ribogreen assay showed, we had the foreign mRNA inside. And we also knew that we needed a more controlled manufacturing process and to adjust the dose for the next step, which is what we did in the refinement of the formulation by switching to microfluidics manufacture and adding a sterile filtration step, as well as adjusting the dose as mentioned. So this is what this workflow illustrates here. This refinement step was only performed on the tetravalent formulation and the blank for reference. And here we only used the liquid forms, as the freeze drying process was being further optimized, or is being further optimized in our lab by another PhD candidate, Aya Halmi. So we might come back to that later. So for this refinement stage, we saw much smaller particles than in the previous step. So here we have particles whose sizes are below 100 nanometers and PDIs are below 0.2. So this can be attributed to the more controlled microfluidics manufacture, but also to the sterile filtration, which traps bigger particles and potential aggregates. For zeta potential, we still had the positive values, and this was coherent since the formulation itself and the lipids used did not change from one step to the other. The ribogreen assay confirmed again that we had encapsulated mRNA concentrations, which were encapsulated mRNA concentrations, proving that we had the 4 mRNA serotypes inside the formulation and a high encapsulation efficiency. And for the osmolality values, they were similar to what was found in the first step, as here for the liquid formulations, we do not have the use of the cryoprotectant. For the biological characterization, transfection efficacy was slightly improved from the previous step, with transfection occurring in both replicates. And the cytotoxicity assays showed that at all the tested dilutions, there were no cytotoxicity for the blank formulation, which we tested. So the perspectives for this ongoing work were a preliminary in vivo immunogenicity study that was conducted recently, with the goal to determine the immunogenicity of the tetravalent formulation in mice, so to assess its humoral and cellular responses, so to check whether it's capable of eliciting high levels of neutralizing antibodies and T-cell responses, specific T-cell responses against each of the Dengue serotypes, and the results are awaited and they are to be analyzed soon. And this will guide the future steps. Either we will need to refine the formulation in case of low immunogenicity, or upscale it if the immunogenicity is encouraging. With that, I come now to the end of my presentation, and obviously this was a team effort, so I would like to thank all of the people directly involved in the project, whose names are here in bold. So from the Chula VRC professors, Ike Chai from Pechara and Chotitorn Ketloy, as well as Dr. Kirayus Kosong from Geneva, my PhD supervisor, Professor Gary Porchard, and also Dr. Olivier Jourdan from our lab, and the 3 master students which have worked, which have done a tremendous work in this project. So Aya Halmi, Amin Rekaibi and Tatiana Bosch da Costa, and also everyone else who has contributed with some help and guidance and technical skills to advance the project. And of course, thank you all today for your attention.
Thank you so much, Alcidia. Great talk. Happy to see that this project is moving forward with a lot of interesting results, especially for the freeze-dried formulation. So I encourage again the attendees to write in the Q&A to ask questions you may have to our speakers. So, Alcidia, I will start with one question that I have. So I saw that you optimized the first formulation, you freeze-dried it, and then you added to your optimized formulation a step of filtration.
Yep.
So I was wondering about the yield. So could you check the yield? Did you have a lot of losses? How did it go?
Yes, surprisingly, it went well. So all of the physical chemical characterization that I showed in the refinement process was after the sterile filtration. So I took a small bit for the characterization, and we did not lose more mRNA from the overall process than we would in the first one where we didn't filtrate them. So that indicates that most potentially mRNA is being encapsulated in the smaller particles, which were not trapped by the filter.
Well, that's a great news indeed. So I have a question for Kanitha. Kanitha, so one of our attendees were wondering what other animal models can do the bactericidal mRNA testing.
May I? What is the question again, please?
So, what other animal models can you check basically for the leptospirosis testing?
The model we have mice and hamster, and we can check the infection only in hamster models because in mice they resist to leptospiral infection. So we can check only immunogenicity of mRNA vaccine in mice. But for the challenge is resist the infection. So we need to test in hamster instead.
Yeah, okay, thank you. Are there any other animal models that could be used apart from mice and hamsters,
For the vaccines, if you want susceptible models, it has been used. For example, tolerable for deficient mice, for example, CTHEJ mice, or some others, for example, gerbills has been reported to use.
Okay.
But the age of the mice and is very important.
Yeah, okay.
Not be too old.
Okay, thank you very much. That's very interesting. Then I have a question for Alcidia from the chat. So, attendees were wondering about your animal experiment, if you examined antibody responses for a long term, and wondered if you test for antibody-dependent enhancements in your assays.
Yeah, that's a great question. So far, this in vivo, it's only a very preliminary one just to assess short-term antibody levels, but challenging the mice with dengue virus and then checking for antibody-dependent enhancement phenomenon is something that definitely will be tested in a later stage if the formulation is encouraging and already elicits high level of neutralizing antibody in the first step.
Thank you, Alcidia, for the answer. So I had another question for Kanitha. So you showed the levels of protections of the two different antigens that you have, and in particularly, AC had a better level of protection. So were you planning also to test ID delivery for that at one point, or you will stick with IM?
Actually, in the future, I want to combine as a multiple antigen, not just one. And we will plan to test intradermal in the future. And we hope that we can use, in general, we should be able to use lower amount of antigen.
Yeah. And I have just a small follow-up questions. Do you see any increased reactogenicity from in the hamster in the ID injection compared to the IM, or not at all?
At this point, we didn't see any difference on the reactogenicity. So, but later we will use the device. Should we get better results of immunogenicity?
I see. Thank you very much. So I will switch again to Alcidia. I have question from the audience regarding the PDI. Asking the reason why the PDI in some of the formulation is higher than 0.2, as would have expected that 0.2 or below would be actually more suitable. Did you observe any impact with this particle size and polydispersity on the immune responses?
So the results for the immune responses are awaited, and for the immunogenicity, we only tested the refined tetravalent formulation and its corresponding blank, so I won't be able to tell that. So we know that for a better immune response, smaller particles and homogeneous or as monodisperse as possible formulations are better. So that's why we try to refine as much as possible. And why do we get these high PDI values in the first place? I'm assuming or hypothesizing it's mostly due to the ethanol injection method itself, which we know is not as reproducible, maybe yielding some aggregates, which, and we saw also in some cases, two different populations before sterile filtering and switching to microfluidics. So, yes.
Thank you. So I had a follow-up question on that. I was wondering, because I saw that you did some TEM images, I was wondering if you did or tried to do any cryo TEM, and in case of that, if you could observe any blebs, or even in the TEM images, could you see any blebs or not at all?
I didn't see any actual blebs in the TEM images, and definitely planning on moving to cryo-TEM as we keep on refining the formulation.
Thank you. So, okay, time is over. So thank you very much for these two great presentations. It was a pleasure to have you both here. We will upload this webinar on YouTube, and I'm sure we will have a lot of views there, and it's going to be very, very successful. So thank you very much for participating and responding to our questions. It was a real pleasure. So, and thank you also to the audience for following and for asking questions. I invite you all to be present to our next webinar, the 7th of May, and have a great day. Thank you very much.
Thank you. Bye-bye.
Bye-bye. Thank you.
Bye.